As a continuation from Quiva, I am now going to discuss next-generation DNA sequencing. An example of next-generation DNA sequencing is Illumina sequencing. I will explain the main steps in how Illumina sequencing works. Step 1. Preparing the genomic sample. The sample DNA which is going to be sequenced is randomly fragmented, and adapters are added to the ends of the DNA fragments. Step 2. Attach DNA to the Illumina flow cell. Due to the addition of the adapters, the single-stranded DNA fragments bind randomly to the surface of the flow cell channels. Step 3. Bridge amplification. To begin the solid phase of bridge amplification, unlabeled nucleotides and enzymes are added to the flow cell channels. Step 4. DNA fragments become double-stranded. The addition of the enzyme incorporates nucleotides to build double-stranded bridges on the solid phase substrate. Step 5. Denature the double-stranded molecules. By denaturing the double-stranded molecules, we are left with single-stranded templates anchored to the substrate. Step 6. Complete amplification. As a result of the previous steps, several million dense clusters of double-stranded DNA are generated in each channel of the Illumina flow cell. We now have enough DNA to begin sequencing. Step 7. Determine the first base. This is the first chemistry cycle. To begin the first sequencing cycle, all 4 labeled reversible terminators, primers, and DNA polymerase are added to the flow cell. Step 8. Image first base. After laser excitation, the image of emitted fluorescence from each cluster on the flow cell is captured. The identity of the first base for each cluster is recorded. Step 9. Determine the second base. This is now the second chemistry cycle. To initiate the next sequencing cycle, all 4 labeled reversible terminators and enzymes are added. Step 10. Image second base. After laser excitation, the data is obtained as before, and the identity of the second base for each cluster is recorded. Step 11. Sequence reads over multiple chemistry cycles. The steps involved in sequencing are repeated to find the sequence of bases in the DNA fragment, one base at a time. Step 12. Align data. It is necessary to align the data from the sequencing cycles and compare the resulting data to a reference to identify sequence differences.